Hey guys, it's Mr. Schmitz. We are back with another lecture. This time we are talking about both nucleic acids, and then we will finish up with proteins. Before this, we talked about carbohydrates and lipids. So nucleic acids and proteins are the other two of the four major macromolecules. So nucleic acids, the monomer of a nucleic acid, which again you'll need to know the monomers for all of these. The monomer of a nucleic acid is a nucleotide. And so a nucleic acid is something that is built up of repeating nucleotides. There are several different types. The three that we're going to be working with are DNA, RNA, and ATP. So deoxyribonucleic acid, DNA, ribonucleic acid, which is RNA, and adenosine triphosphate, which is ATP. So to make up a single nucleotide, we have three components. We have a nitrogen base, or nitrogenous base. You might hear it said either way, nitrogen base or nitrogenous base, that's the same thing. But a nitrogen base, that just means it contains nitrogen. We have a 5-carbon sugar. You might hear that called a pentose sugar. And you have a phosphate group. So these three things combine to form what is called a nucleotide, which is the building block of a bigger nucleic acid. Nucleic acid's job is they're going to store and transmit hereditary information. So your genetic information, if you remember back to the characteristics of life, containing genetic information is one of those characteristics. Your DNA in humans is going to pass down your genetic information. So we'll start with DNA. DNA is deoxyribonucleic acid. We're going to talk about DNA a whole bunch over the year. So this will just be a brief overview of DNA. But DNA is double-stranded. As you can see here, you have one strand on the left and one strand on the right. Here would be one single nucleotide with a nitrogen base, a sugar, and a phosphate. That's one single nucleotide. So you have 1, 2, 3, 4 nucleotides on the left and 1, 2, 3, 4 nucleotides on the right. It's made of a pentose sugar that is called deoxyribose, which is where deoxyribonucleic acid gets its name. So this 5-sided sugar's name is deoxyribose. The only place that we find DNA is inside of the nucleus of cells. The nitrogenous bases that I mentioned, there are 4 nitrogenous bases in DNA. They are abbreviated A, T, G, and C. Those stand for adenine, guanine, cytosine, and thymine. And it is anti-parallel in construction, meaning they go in opposite directions, the two strands do. So the easiest way to visualize this is if you look at the orientation of the sugar. You have the point of these sugars facing up. You have the point of these sugars facing down. They're oriented in opposite directions. This brings us to RNA. RNA is ribonucleic acid. It is made of ribose sugar instead of deoxyribose sugar. So that's where RNA gets its name. Its sugar is different. It is single-stranded instead of double-stranded. And it can go outside of the nucleus. So it actually will go inside and outside the nucleus, but it can go in and out of the nucleus. It has A, G, and C, just like DNA does. One difference, though, is that it does not have thymine. Instead, it has uracil. So there's one base that is different, and that is uracil. And RNA has 3 types. It's got mRNA, rRNA, and tRNA, which stand for messenger RNA, ribosomal RNA, and transfer RNA. Each one of those does a separate job in the cell. Messenger RNA, as it sounds, takes a message. It's going to copy a DNA strand and take it out of the cell. Ribosomal RNA, as you can see pictured here, creates the ribosome that we think of, which we will talk about during the cellular unit, but it's going to be responsible for making proteins. Transfer RNA is this molecule that is bringing that little blue dot, which is an amino acid. It is going to bring amino acids to form a protein. So all of these RNA are involved in the process of making proteins, in protein synthesis. Which brings us to proteins. So, proteins. The monomer of a protein is called an amino acid. The structure of a protein, if you can see this middle part, it's got carbon, nitrogen, oxygen, hydrogen, and then this R group. Now, R is not an element. Okay, R is just a placeholder for us. But this general structure you will need to know for proteins. They have an amino group that is NH2 in this box here. Every protein, every amino acid has this amine group. And then you have a carboxyl group, which has COOH, COOH. And then you have the R group. The R group is the only difference between amino acids. So this is, like I said, a placeholder. Some structure of elements, some compound will be here that will give the amino acid its unique properties. R is just a placeholder. Like I mentioned, they're going to contain carbon, hydrogen, oxygen, and nitrogen. So, amino acids. We have 20 different amino acids. Some of them have charges on their chains. Some of them are polar. There are some very unique ones. Some of them have hydrophobic chains. But there are 20 different amino acids. We sort of think of these 20 amino acids like an alphabet. And it's going to be these different amino acids or the different letters that get put together to make the words that the proteins are. So based on which amino acids we grab and put together, we're going to create a different end product of protein. So protein has several different types of structures. It has a primary structure. That is the first structure. A primary structure is just a straight chain of amino acids. So if I go back and I grab 4 amino acids, that makes an amino acid chain. That chain is considered its primary structure. The secondary structure is when we fold or coil that chain. So that forms two options, either a pleated sheet, which is this zigzag, or a alpha helix, which is the helical coiling structure. So you've got the zigzag pleated sheet or the coiling alpha helix as your secondary structure. So this chain of amino acids begins to bond with itself, and that's why that occurs. So you see it start to either coil or pleat. The tertiary structure is the three-dimensional arrangement. So when a pleated sheet and alpha helix begin to cross over themselves, they form a tertiary structure. That's typically what we think of. So we've added maybe multiple alpha helixes together with pleated sheets, and you can now see that they are going around and over top each other, and they are crossing each other and coiling and pleating. That is your tertiary structure. And then finally you have what is called the quaternary structure. Quaternary structure is when you take multiple tertiary proteins and put them together. So you see one blobbed protein, and then in the quaternary structure you see 4 polypeptides. So you have the protein structure from primary to secondary, secondary to tertiary, and tertiary to quaternary. Proteins have a ton of functions and applications in our body. The easiest way to think about it is anything that is being carried out in your body is probably being done by a protein. So some examples of that are enzymes. Enzymes we're going to focus on. We'll actually be doing a lab on them. But enzymes, one example of an enzyme is amylase. Amylase breaks things down and helps with digestion. We have proteins that help in transport. The protein hemoglobin carries oxygen in our blood. We have proteins that help store. We have proteins that help us contract and move our muscles, like myosin and actin. They're what allow you to move. We have proteins that provide protection, things like antibodies that fight infection if something gets in our bloodstream. We have hormones like growth hormone that help you to grow and mature. We have insulin that helps you metabolize carbohydrates, so break down carbohydrates. We have structural proteins like collagen that help build our tendons and bones and cartilage. And we have toxins like ricin, which is a toxin that can be poisonous. And snake venom is made from proteins. So as you can see, proteins have a ton of different functions. You should know some general functions and maybe some of the common examples of proteins, like myosin and actin and hemoglobin. But I'm not going to make you memorize every single one of these examples. So that's going to end our lecture today over lipids and proteins. Excuse me, nucleic acids and proteins. Thank you.